So this is the final presentation of group FOH and the function of our unknown gene.
Trying to determine the model of our unknown gene, we utilized the homology-based method as it allowed us to compare our sequence of transcripts from other model organisms, giving us more accurate results. Using the Phytosome 12 software, we determined our gene, Brady5G15190, contained 5 exons, was located on the 5th chromosome, and coded for a peptide sequence 733 amino acids in length.
We also determined the protein domains located in our gene of interest using Phytosome 12, and as you can see from the figure, our gene of interest is part of the trichome bifringins like 2 family and possesses a PC esterase and PMR5N domain. The PC esterase family has been associated with modifications of the cell wall glycan properties, as well as xylan acetyltransferase activity, and even plays a role in freezing stress. Similarly, the trichome bifringins like 2 family plays a role in the acetylation of cell wall polymers and also plays a role in freezing stress.
To determine how well our protein was evolutionarily conserved, we constructed a phylogenetic tree which compared our sequence with similar sequences belonging to other plant species. We determined that our gene was well conserved. Our protein of interest is highlighted here in green, and you can see its most recent ancestors shared with Oryza sativa, and next most recent ancestors shared with Solanum lycopersicum. We could also determine that our protein is closely related to Arabidopsis thaliana. Since our phylogeny indicated that our protein of interest was most closely related to O. sativa, we looked in the literature for trichrome birefringence-like 2 proteins located in O. sativa. In one study, loss of function mutants for trichrome birefringence-like 2, the plants demonstrated a reduction in monoacetylation of the xylan and therefore experienced collapsed xylem vessels, as well as stunted growth, pathogen defense, and cold tolerance. Since these changes in phenotype occurred in plants without trichrome birefringence-like 2 proteins, it can be concluded that such proteins aid in acetylation of xylan, growth, disease resistance, and cold tolerance. In our literature search, we also looked at the closely related Eskimo-1 gene, which shares the same protein domains as Brady5G15190. In a study, it was found that mutations in Eskimo-1 resulted in significant reductions of xylan acetylation and stem mechanical strength. It was also found that Eskimo-1 mutants had collapsed xylem vessels and acted as a negative regulator of cold acclimation. Therefore, it can be concluded that Eskimo-1 plays a role in these functions, and it can be inferred that our protein has similar functions because of the shared domains.
So as you can see from our gel electrophoresis on the left, we were able to successfully extract DNA from our mutant and control plant stocks. However, the DNA ladder did not run correctly, so it's difficult to determine the exact length of DNA we extracted. Although our DNA was determined to be of adequate concentration and purity assessed with the nanodrop device, we were unable to amplify our DNA through PCR. As you can see in our gel electrophoresis, there is no DNA bands in either of our PCR experiments. In the first gel, we don't have a DNA ladder, so we ran in well 4 of the middle gel, and you can see that there's no DNA. And in our second PCR amplification, we also did not see any DNA in our gel.
We attempted to make qualitative observations of the cell wall composition of tissue from the first nodes of mutant and wild-type plants. We were unable to draw any significant conclusions because our plant stocks were dried out and the structural integrity was compromised. As can be seen in our figures, our cell walls and vascular bundles were not all intact, making it difficult to determine differences between mutant and wild-type. Based on advice from our instructor, quantitative analysis was not performed. We expected to see changes in xylem composition because our gene plays a role in xylan acetylation as well as maintaining the structural integrity of cell walls. We could not conclude there were structural differences in the xylem.
So as you can see from the pictographs, the gene expression of our gene of interest is highly expressed in the roots, young grain, and young seedlings. This high expression may indicate that our gene plays a role in growth and the strengthening of growing cell walls. The moderate gene expression in the stem can also support this idea as the stems are continuously growing. Additionally, since the roots are heavily involved in water transport, the high gene expression could indicate that our gene of interest may be involved in water stress, which was previously indicated in our literature review.
Based on data from the EFP browser in the Hazen lab, it seems our gene's expression is controlled by thermo and photocycles. Looking at the pictograph, gene expression is increased after cold conditions. This is supported by the p-value of our t-test between light, dark, hot, cold, and light, dark, hot, hot, which was less than 0.05, showing a significant difference in gene expression. We also performed a t-test between light, dark, hot, hot, and light, light, hot, hot, in which the p-value was also less than 0.05, indicating a significant difference.
To test if our gene was actually involved in water status, we performed an experiment in which 3 plants were exposed to a natural drought for 5 days and 3 other plants were exposed to control settings and watered regularly. We extracted RNA from the leaf and stem tissues of both the control and experimental groups. We accidentally discarded the supernatant for control 1 and 3, however, so we do not have the RNA samples for those biological replicates. For the samples that we were able to extract, we concluded that the concentrations were adequate, even though the experimental sample 3 and control sample 2 were diluted due to a pipetting error. As you can see from the gel, we were able to successfully extract RNA as seen by the 2 bands in each of the wells.
With the RNA we extracted, we were able to construct cDNA to run qPCR with. Since we did not have control 1 or 3 samples, we used wild types 1 and 3 as a substitute based on Ahmed's instructions. Our control plants were regularly grown in soil, so it made sense that we could substitute wild type samples for our control samples. When we received the qPCR results, we were missing data. Specifically, we were missing both technical replicates of control 3 with the gene of interest primer, so it had to be excluded from our analysis. There was a large variance with controls 1 and 2, as well as the experimental groups. We conducted a delta-delta CT test to determine the average fold change in the control groups and the treatment groups. And as you can see on the graph, there was a large difference in fold change between the 2 groups. When we ran the t-test, however, the p-value was returned as 0.052, indicating no significant difference between the 2 groups. We suspect that the variance in the data could have led to the high p-value. More analysis would need to be completed to determine the differences.
So overall, the key takeaway is that Brady 5G 15190 is likely involved in cold tolerance, water stress, growth, disease resistance, and acetylation of cell wall polymers. While the literature suggests that Brady 5G 15190 is involved in water stress, our experiment with natural drought showed no significant differences in gene expression levels of our gene of interest. So more research would need to be conducted to actually conclude if our gene is involved in water stress. Thank you for listening to our presentation, and we hope you enjoyed.